high-fat mouse had less vasorelaxation compared to the normal diet mouse. And here's another test to test the same thing. So here what you do, this drug called PE or phenylephrine. So if you contract the vessels, now you contract it with this PE, you put PE, it just tightens up. And then you allow it to relax. So the better it relaxes, it means more of nitric oxide is present. So in here, you see the control mouse, they relax. After you contract it, you allow them, they relax. You know, like you blow a balloon and then allow it like that. So you contract it and then it relaxes here. But high fat didn't relax as much. It was significantly lower. So we do all these statistical tests. You repeat it several times and see if you have a consistently stable, or you have a repeated measure showing you the same thing. Okay? So we know that in the high fat fed mouse, they have less nitric oxide. They are having some vascular dysfunction if the vessel is not relaxing as much. And this one, this drug called as sodium nitroprusside, you throw it on the vessel, you take an intact vessel, right? This one directly, it bypasses the endothelium and affects only the smooth muscle. So it causes smooth muscle relaxation. In this case, you can see both high-fat fed mouse and control-fed mouse have the same effect, meaning smooth muscles are not at all involved in this process. It's all endothelium. So that's like a control to test, which layer is affected. So you bypass the endothelium, it doesn't have an effect on the endothelium, it just goes to the smooth muscle, and there it relaxes it. Control and high-fat fed mouse both relax at the same time. I mean, they were fine. The smooth muscle is fine, but not the endothelium. So the effect is in the endothelium. So that's our conclusion. Are you guys clear so far?
Yeah.
Do you have any questions? You get it, right?
Waiting to see.
All right. I know this may be a little off your thing, but I hope you just get the big picture. I know what happens. Nitric oxide.
No, when we talk about diet, this is actually going to be really helpful when we talk about diet and why types of fats matter so much, because there's been a lot of media coverage of fat in the diet, and all fats are basically conflated together, and we made mistakes, right, in nutrition epidemiology in terms of recommendations, but we've been paying for those on the other end now. So this is actually really helpful when we talk about the mechanisms of why that's the case, because you're going to have lots of people saying, oh, my CrossFit gym says I should eat all coconut oil, and now you're getting some of the tools to understand why that's not nutritionist recommendation.
So, now what we were thinking is, so what's different about this free fatty acid? What's happening? What component of the free fatty acid could be influencing in us? Once again, we searched the literature and trying to find what happens, how is the fatty acid broken down, right? We constantly saw something called as ceramide. It is a lipid, so when you eat your fatty food, saturated fats, they get pretty broken down, and the final, you'll see the pathway, I'll just show you a pathway briefly. So the final product of that free fatty acid is a sphingolipid known as ceramide. And ceramide accumulates in the arteries, it accumulates in the liver. Right? So we wanted to see if our mice are also having that kind of phenotype. And we saw definitely in the arteries and the liver, we saw high-fat fed mouse having excess ceramide. So what is ceramide then? So if you have high free fatty acid, it's a breakdown pathway, how food gets broken down in your body. So you have palmitic acid or palmitate, that's the predominant breakdown product of C16, breakdown product of your high-fat diet. It goes through an enzymatic pathway, and you have important enzymes like serine palmitoyltransferase, which is the start or initiator enzyme, and then the pathway eventually leads to something called as a ceramide, the toxic sphingolipid. So this pathway, there's another important enzyme called as dihydroceramide desaturase. So the pathway just goes up to here until here it's kind of inactive. It's not bioactive, just breaks down. But when you gets to this place, it becomes a bioactive lipid. So there's bioactive lipid and inactive lipid. So the lipid might be there and not having, or you didn't find it yet. So it means that there, right? So that kind of, these are not bioactive, but once it processes, it becomes an active molecule, meaning it can signal, it can bind, it can do all kinds of stuff. So how do we study the, okay, next question is, if ceramide, we saw this ceramide being, accumulating all this. So if ceramide accrual is prevented, if you could stop that ceramide from accumulating, can this improve nitric oxide? Or in other words, is ceramide the cause for the destruction of NO? So that was the question. So if yes, then how does ceramide regulate nitric oxide? So this is when we got into the biology of it. Okay? So here's the pathway. How do we stop ceramide from happening? How do you stop? You stop the enzyme that breaks it down. So you can break down this serine palmitate transferase, the first enzyme that results in the conversion of palmitate, the initiator of this pathway. You can stop it with a pharmacological agent called myriocin. It's from a fungus. It's a product of, from a fungi. So that is a pharmacological agent, right? And then you can do a genetic disruption. So these two are genetic disruption. So genetic disruption is, we have the DNA, the information from the DNA is copied onto an mRNA, right? Your molecular biology class, DNA to have DNA. And then information is copied onto mRNA strand. Then mRNA goes to a process of protein synthesis to result or to make the proteins, right? So if you want to, these are all proteins, enzymes are all proteins. So if you want to chop one protein or stop an action of an enzyme, what you do is, there are small strands of RNA that you call as siRNA, or you call it as small interfering RNA or silencers, and you throw it into the cell. These silencers will go and bind to small segments of this mRNA. That will basically mess up the protein synthesis. That small sequence that binds it distorts its structure. It doesn't go into protein synthesis anymore. So small siRNA technology is great, and we use it all the time to chop off protein. If you want to look at a function of a protein, throw in siRNA and then stop its synthesis and see what happens in the cell. That's a technique you do routinely in our labs. And another same technique, you can knock out genes in a live animal. So you can take an animal and knock out a gene. For example, in this case, we stop this dihydroceramide desaturase enzyme from becoming a protein. We just stop its synthesis. So you can stop this and you can stop this. So that's the path we took. Stop the first step, or stop the step where the inactive form is converted to active form. See what happens to mouse. Okay, clear so far? So next, what we did. Now, we have the same mouse model. We have two more groups. So how we stop here, just trying to tell you, this one, DES1, is that enzyme dihydroceramide desaturase. So DES1. If I say DES1 plus, meaning it's just a regular mouse. It's got the full enzyme, active enzyme. DES1 is completely active. DES1 plus is a mouse that is just wild type, normal mouse, no knockdown here. DES1 plus minus is called a heterozygous mouse. I'm not going into the genetics of it, but think of it. So we chop the enzyme, so it has less amount of the enzyme dihydroceramide desaturase. The enzyme is less here, so the knockout mouse has about only half of the enzyme that is present. So it cannot convert actively whatever dihydroceramide is synthesized. It will be like less active. You have less of ceramide in these mice. Now you look at the model. So arterial ceramide accumulation, control mouse, high-fat mouse, ceramide accumulates. But in the knockout mouse, this one mouse, ceramide accumulation is prevented. So your knockout worked. You stop the enzyme, the mouse is not able to make more of ceramide. So model works again. Look at the body composition. So Desmond mice, they were on high-fat diet too. You look at it, the lean body composition, fat mass and lean mass, we looked at the mice, fat mass and lean mass, they are on a high-fat diet. Lean mass,
Do they have a little bod pod for mice?
so like NMR, mouse NMR, so we did that, and we saw they are as fat as the other mice, right? Because they are eating a high-fat diet. The only difference is you're not having ceramide here, but they have a less lean mass, high fat mass, so they are fat mouse, so they don't have arterial ceramide. And then when you look at GTT, they are as bad or even worse. They seem to be worse in the sense we are still trying to figure out why it's so worse. So they are unable to clear glucose from their system. So the high-fat diet is having the same type of influence in them. But they don't have ceramide accrual because you stop ceramide synthesis. Now you look at vasorelaxation in these mice. Vasorelaxation, an indicator of whether nitric oxide is produced or not. You're trying to link arterial ceramide to eNOS. If ceramide is the cause for this eNOS, lack of eNOS bioavailability. Now if you see here, this is wild-type mouse. High-fat mouse had less vasorelaxation. But if you look at DES1 mice, the mice that does not have ceramide, the vasorelaxation is basically similar between the control and high, although it was lower, but there was no difference between a control and a high-fat fed DES1 mice. They seem to be.
Is that still in the normal range? Because you see the control
Still in
mouse
the-
is a little bit lower than it was when
Yeah.
you didn't knock out the gene, but it's still normal
Still
function.
normal function. Yeah. And if you see eNOS phosphorylation, you see that's high-fat mice, you have very less of eNOS phosphorylation. In the DES1 mice, you start getting a little bit more. You still have more of eNOS in this cell group. So there's a connection between ceramide and eNOS. Even though the body fat mass was very similar to a wild-type mouse on a high-fat diet, even though it was as resistant to, or it was becoming insulin resistant and developing impaired glucose tolerance, the only difference is ceramide here. Ceramide was gone. You seem to get better eNOS function. eNOS is coming up. So there is a link. So inhibition of ceramide accumulation prevents high-fat diet-induced arterial dysfunction. So we know that if you inhibit ceramide, even though everything else is the same or worse, you still are able to recover eNOS. So there is a link between ceramide and eNOS. So how does ceramide regulate eNOS? Now we have to do the mechanism. So if you have to do the mechanism, you have to have a cell culture model. So just for the ease of it, you have to handle cells and you have to do all these molecular mechanisms. You can't be killing like tens and thousands of mice for each assay, right? So you just get important information from the mice, and then you can go back to your cell culture system and evaluate the mechanism, then test the important endpoints in the mouse, and then if everything works, you can test it in human beings. If it's ethical and all that, then you can try it out in human beings, right? So what we had to do, we moved to a cell culture model. What is a cell culture model? We have these endothelial cells, right? So we just get those cells and plate them in a petri dish, a round petri dish with media in it. So we have the endothelial cells in a petri dish, and you can add stuff to it and study its molecular mechanisms. So how do you do? Endothelial cells, high-fat feeding. How do you feed endothelial cells a high-fat diet? You basically throw in palmitate. Because you eat that food, breakdown product is palmitate, predominantly palmitate. So you get a commercially available palmitate, and then you treat the endothelial cells with palmitate and see what happens. What happens to ceramide. So first thing we want to see here, once again, that is vehicle, meaning the untreated one. You're not having any high fat in it. The next one is palmitate treated cell. You're looking at ceramide accrual. Once again, this is a technique. So if you put a fluorescence molecule into the cells and it will bind to palmitate. If you have more palmitate, the fluorescence is more. If you have less palmitate, the fluorescence is less. So you vehicle, no palmitate, no ceramide, it binds to ceramide, and it fluoresces. So palmitate treated cell had increased fluorescence, meaning it had more ceramide accrual. Now if you see the previous slide, you had this Meriosin. It's easy to treat cells with the drug Meriosin. And we did silence with this knockdown too. You'll see the same data down. So here is the knockdown. So this is something we do to confirm our efficiency of knockdown. That is the protein production. If you use that siRNA, you have less of protein production, meaning you are successfully knocking down that protein expression. So in these cells, scrambled siRNA, you have a mimic of siRNA, but it doesn't really knock the protein out, so that serves as a control. And then you have the siRNA. Now you look at ceramide accrual, you see very less when you silence that gene responsible for ceramide synthesis. So ceramide accrual was successfully prevented in cells treated with either SPTLC siRNA or Meriosin, similar to DES1 knockout in the animal model. So you are preventing ceramide. Now you are going to look at eNOS. So this is a bit complex. If you see, this is basal, and that is after insulin, and this is after palmitate treatment, basal and insulin. So it's very blunted compared to just insulin and basal, right? When palmitate treatment occurs, you have less of eNOS phosphorylation. But Meriosin restores it. So you have Meriosin restores. What does Meriosin do? Meriosin inhibit ceramide accrual. When ceramide accrual is prevented, you see eNOS phosphorylation being restored. So once again, cellular connection is made, right? So now we went to an arduous process of figuring out what's ceramide doing, how is ceramide bringing this, right? So nitric oxide synthesis, or the bioavailability, depends on its synthesis versus its destruction. So destruction can occur when reactive oxygen species is present, which is hydrogen peroxide, superoxide. If your cell, for example, high-fat diet produces hydrogen peroxide. If you eat a high-fat diet, you will have more of reactive oxygen species, because breakdown of that fatty acid in the mitochondria generates more of reactive oxygen species. Reactive oxygen species can bind to nitric oxide and can form another species called as peroxynitrate. So peroxynitrate can destroy nitric oxide. So we wanted to see, high-fat diet, this person eats it or the mouse eats it, does it form more reactive oxygen species, and is that the reason why does ceramide generate more reactive oxygen species, and is it why nitric oxide is prevented? But it was not the case. We saw more reactive oxygen species. It was not really binding and preventing NO. So that was a long
Back to the
process.
drawing board.
Yeah, so we didn't have, that was not the reason. So we had to leave that. Then, so as I mentioned earlier, you know protein phosphorylation and dephosphorylation, right? So these are very few of the kinases that can phosphorylate eNOS. Very minimal of them are represented here. There are a ton. So these are common kinases that phosphorylate eNOS. We looked at them, they were all phosphorylating normally. AKD was fine, AKD was phosphorylated, but we were wondering what's happening. Why is it's phosphorylated, but it's not phosphorylating eNOS? But they were fine, so we had to eliminate defective upstream signaling. But the next option is, we want to see if there is a phosphatase that is taking off phosphate group or inactivating. Sometimes phosphatase, their activity can inactivate eNOS. So is there a protein phosphatase? Kinases are fine. What about phosphatases? So we searched the literature to see what's the common phosphatase, what's the most abundant phosphatase that could be in an endothelial cell. We came up with this phosphatase called as protein phosphatase 2A, so that is PP2A. So So protein phosphatase 2A activity shot up so much when you treat the cells with Palmitate. So that's an indicator that we thought, oh, maybe here there's something that we are striking. Maybe it's the phosphatase. Why is it activity that high? Then what we did, so this is a drug called as, the drug has not even gotten its real name. The company that makes it is called as Lixti Biotechnology. So it's got a name LB1. So this drug is actually in clinical trial for cancer, lung cancer treatment. And this professor contacted us and said, oh, I have a phosphatase inhibitor and it's been working well with clinical trials. Do you want to try and inhibit the phosphatase in high-fat mice, right? We are looking at obesity, it's got links to metabolic diseases, cancer, so he was interested in partnering with us and he provided us some LB1. So we took that LB1, which is a phosphatase inhibitor, and we threw it in cells with palmitate, and you can see how PP2A activity is done. So that drug it inhibits the activity of PP2A. So this is another way of testing whether PP2A activity is down. Once again, it's a Western blot. This is a direct measure. Activity assay is gone down. In this case, this phosphatase, if it is dephosphorylated at this site, means it is active. It's the other way around. You don't have to understand that, you're just letting it. It's another way to test if the activity is up or down. And here, when you treat with LB1, the phosphorylation increases, meaning its activity goes down actually. So the activity is gone down. Treat with LB1. Now, if you look at enos phosphorylation, palmitate-induced decrease or reduction in enos phosphorylation is restored with LB1. Meaning, if you either prevent ceramide, you can restore the activity of enos, or if you prevent the activity of the phosphatase LB1 or phosphatase PP2A, you can restore the activity of enos. So either ceramide or the phosphatase PP2A or together are acting in preventing enos activation. So then this is the actual measure of nitric oxide. Palmitate decreases NO. You've been seeing enos, right, the enzyme activity. Now this is an actual measure of nitric oxide itself. So nitric oxide is low with palmitate. We treat with LB1, which inhibits PP2A activity, enos activation or nitric oxide reduction is restored, which will result in vessels relaxing more, right? So how does that do? And we saw this AKT-enos interaction, right? I'm just showing it to you as an image here. So enos, AKT sits with enos and phosphorylates it. And this HSP90 is a protein that acts as a chaperone or a holding or somebody that keeps them together. So PP2A somehow binds to enos and dephosphorylates enos. So you're wondering, is it dephosphorylating enos? Is it dephosphorylating AKT? Somehow it's removing the phosphate group from it. How is it doing? And LB1 is preventing it. What is the mechanism behind it? So we wanted to test it in a mouse model. So another reason why we want to test it in a mouse model is to see if this LB1 drug is toxic to mice, right? You can make a drug, but if you inject it into a human being or a mouse, and if the mouse dies, right, you don't really know, you don't really have anything there, right? The drug killed the mouse, right? So we injected into the mouse, like it was a 21 days or 12 weeks of high-fat feeding, and the last 21 days we injected the mice, a group of mice with the LB1. So controlled high-fat, that's how we check the activity of PP2A. If it is less phosphorylation, it's more active. It's a reverse of enos. So controlled high-fat fed mouse, you have increased PP2A activity. When you inject with LB1, the activity is gone down. More phosphorylation, but activity is gone down. So we know that the mice had, when you inhibit PP2A activity, or the LB1 drug is efficient in inhibiting PP2A activity. Now, we had a suspicion based on the previous, this kind of interaction We wanted to see really physically enos and PP2A interact. Are they sitting together in a high-fat diet? So there's a technique called as immunoprecipitation. So you pull out one protein and then you see what all is coming along with it, right? It's Western blotting or immunoprecipitation followed by Western blotting. You pull one protein and you see, for example, if I have a clip here, I attach it or I try to pull, so I pull enos like that, and then something else comes with it, like that, so you know they are physically interacting with each other. So we pulled enos and we want to see who is sitting with it, or we did it, I'm showing you a reverse because we did all kinds of combination. We pulled PP2A and then blotted for enos, and enos came along with it, stuck with it. So we know they are physically together. And if you see high-fat diet, PP2A, more of PP2A, or have more of PP2A association with enos. And you give LB1, here's when we thought, oh my God, we don't have the answer here, because with LB1, still more of PP2A was coming with it. And we were like, LB1 is supposed to prevent this interaction, but PP2A is still attached to it. What's going on? And that took another year for us to solve. So it was just sitting with it and coming along and pulling out all the time. And once again, if you IP enos, we wanted to see the whole complex, whether AKT associates with it. So you pull enos and you have AKT association. But with high-fat diet, we had less of AKT phosphorylation there, less association. And we were wondering why that's happening. Same way with HSP90. So when PP2A pulls with enos, everybody else is less. We have less of AKT, less of PP2A, more of PP2A sitting there. So that's dephosphorylating AKT and preventing HSP90 from attaching. So that whole interaction is messed up because of PP2A. Right? And with, again, enos phosphorylation is low. When you treat with LB1, enos phosphorylation is restored in spite of PP2A sitting there. So what happens is we later on figured out that this physical interaction, although PP2A is sitting there with enos, its activity is alone spoiled by the drug LB1. Say the protein is still holding on, but it's like a dead protein sitting there, not doing anything. Or its hands and legs are cut off, it's just stuck to the enos and not doing anything at all. So even though the LB1 was not efficient in pulling them apart, just left them there, but it's not functional. The protein phosphatase was it was like without any function, right? So that actually restored enos activation. Its activity to take off phosphate, being a phosphatase, is gone in LB1. So now we looked at the same group of mice and their vasorelaxation. So endothelium dependent, you put the drug acetylcholine, it acts directly on endothelium. And you see with the high-fat diet, you have less of vasorelaxation, but LB1 restores it. You see that 3 different doses, just went with higher and higher dose and saw or lower and lower dose, and we saw how well you can restore enos phosphorylation or nitric oxide production, and your vascular function improves. Smooth muscle, you use the other drug called sodium nitroprusside, no change, no difference. Once again, pointing out the endothelium is the culprit, and everything is happening in the endothelium, not the smooth muscle, right? So that shows LB1 restores vascular function. LB1 inhibits the activity of PP2A. So we know now that PP2A is involved in destruction of enos by its dephosphorylating activity. Either dephosphorylate AKT or dephosphorylates enos. It causes the effect, of less enos activation, less nitric oxide production. If you prevent the activity of PP2A with LB1, then you can restore it, or restore enos function. So why we were talking about ceramide and why PP2A? Ceramide accrual somehow activates PP2A. How does it do that? PP2A, so this is a conclusion, right? Basically a summary of what we saw so far. PP2A associates with eNOS, disrupts the interaction between AKT, HSP90 and eNOS. And this disruption is prevented if you either inhibit ceramide with myriocin or PP2A